تمام أو بدأت.
Okay. So, hello everyone. Firstly, I would like to welcome all attendees in MGBS 2024. Under patronage of Dr. Ahmed Al-Minshawi, the President of the University, Prof. Dr. Gamal Badr, Vice President for Graduate Studies and Research, Dr. Adil Mahmoud, the Dean of Faculty, Dr. Mohamed Al-Saghir, Vice Dean of the Graduate Studies and Research, Dr. Baha El-Din, the Head of the Genetic Department. It's our pleasure to start the first lecture in MGBS 2024. In this MGBS, a total of 12 lectures will be given this year. The lectures will cover important topics in the field of molecular genetics and biotechnology in humans, plants, animals, and insects. You will find all information in our website. The first seminar today will be given by Prof. Dr. Mohamed Ahmed Ibrahim, a professor of Plant Protection, Faculty of Agriculture, Asyut University in Egypt. And let me please introduce him in few words. Dr. Mohammed graduated from the Faculty of Agriculture, Department of Plant Protection in 2000 with excellence. He got his master from the Department of Plant Protection, Faculty of Agriculture. Then he moved to USA to get his PhD from University of California, Davis. Dr. Muhammad won the award for the best research in agriculture sciences for the year of 2021. Dr. Muhammad has more than 65 research papers published in high international journals. For example, a Journal of Hazardous Material. He is a member of editorial committee of Frontiers in Environmental Science, Journal of Saudi Society of Agricultural Sciences, and Food and Chemical Toxicology. Dr. Muhammad is a principal researcher for many local projects affiliated to the Egyptian Ministry of Agriculture. We would like to thank you, Dr. Muhammad, for accepting our invitation and filling the application to present your outstanding research in MGBS 2024. And please start mic with you.
Thank you, Dr. Ahmed. Hi, everyone. This is Dr. Mohamed Ahmed Ibrahim, Professor in Plant Protection Department, Faculty of Agriculture, Asyut University, PhD from University of California, Davis, United States of America. At the beginning, I would like to thank Dr. Ahmed Atif Salam, the founder of the MGBS. And today, actually, this is my 5th lecture I given to the MGBS. And big thanks to the organization committee too. Today, I would like to give a lecture on gene expression profiling in liver of zebrafish exposed to ethylhexyl methoxycinnamate and its phytoproducts. The contents will be introduction, general hypothesis, material and method, results, and conclusion. At the beginning, actually, sun release two types of rays. The first one called UVA, which can penetrate your skin more deeply and cause your skin cells to age prematurely. About 95% of the UV rays that reach the ground are UVA rays. The other 5% of UV rays are called UVB, which is the second type of ray. They have higher energy levels than UVA rays and typically damage the outermost layer of your skin, causing sunburn, and that's why we use sunscreen. However, without using sunscreen, 100 photons enter the skin by using the SPF 15 sunscreen that will give 90% of protection, and 7 photons enter your skin. Whereas by using the SPF 30 sunscreen, that will give you around 97% protection, and only 3 photons enter your skin. But why do we need to use the sunscreen every day? First, sunscreen is very important for reduce the risk of sunburn. Second, prevent the signs of aging. Third, reduce your risk of skin cancer. Fourth, prevent skin discoloration. And finally, reduce inflammation. But the unwise and huge uses of sunscreen pollution can have major impacts on corals, fish, mammalians, and the whole ecosystems. Not only can it bleach or deform corals, but it can also impair the growth of green algae, which is an essential part of the food web. The effects can be also seen all over the way upon the food chain, even affecting at-risk species like fish and turtles. Unfortunately, of course, through water sampling around coral reef sites, international agencies and organizations have detected high concentration of some of these harmful chemicals, such as ethylhexyl methoxycinnamate, 2-ethylhexanol, and 4-methoxybenzaldehyde, which is a type of photoproducts of the sunscreen. Our general hypothesis in our study is like this. Is there any impact of ethylhexyl methoxycinnamate and its photoproduct on the studied animal models? So this is our question, and we try in our research to give answer of this question. Aim of this study is to investigate the impact of EHMC and its photoproducts 2EH and 4-MBA in zebrafish through molecular transcriptional changes of. We have actually two types of family genes here, stress responsive genes like HSB70, NRF2, CYB1A, and AHR. The second type is antioxidant genes like SOD1, SOD2, CAT, GSTP1, GBX1A, GSS, and GSR. The methodology of our experiments in this study, we try actually to give why we are using zebrafish in our study. Zebrafish has many advantages to use, and zebrafish provide multiple significant advantages over mammalian vertebrate models organisms such as mice and rats. The first advantage, the early development of zebrafish embryo in external rapid and visually accessible. The second one, the embryo and crown. The third one is the protective membrane surrounding the embryo are transparent, which is allows the scientist to follow development from the first moment of fertilization through to free swimming, actively feeding larval fish. Baby zebrafish hatching after 2 days after fertilization and begin actively feeding 5 days after fertilization. During the first 5 days, all major organ, and this point actually really important because the first 5 days, all major organ system develop and begin functioning. Zebrafish are able to breed year-round with females capable of producing several hundred embryos each week. The abundance of offspring is especially advantage for genetic mapping studies and experimental that demand large sample size. And finally, zebrafish can raise and maintain a high-density aquarium system requiring much less space and cost than the animal facilities necessary for mammalians. In this study, actually, we're using three parent compounds. The first one is ethylhexyl methoxycinnamate, and the abbreviation is ELMC. The second one, 2-ethylhexanol, E2EH, and the fourth one actually is 4-methylbenzaldehyde, 4-MBA. The model system and experimental design, wild type adult of zebrafish more than 5 months of age were maintained in the laboratory condition, and the fish were housed in a glass aquarium at 26 degrees, which with the photo period of 12 to 12 hour dark and light, and feed 2 times a day with commercial fish feed. Zebra fish, we're using two sexes, the male and female, ratio 1 to 1 adults, were randomly selected and distribution in 4 experimental groups. The control group was maintained without any compound, and the other 3 groups were exposed to the respected pollutant, the 3 products, the 3 chemicals we tested in this study, at a concentration of 3 microgram per liter. Each group contains 12 fish in a 5-liter exposure tank, and the exposure was performed for 21 days. The control group was maintained separately without pollutant and used only in the corresponding volume on the vehicle, which is used to prepare the stock solution. The experiment was conducted in a semi-static condition and was renewed daily to maintain the concentration of the pollutant. At the end of the 7th, 14th, and 21st days of the exposure period were insufficient in ice, and then liver samples were carefully dissected out and stored in minus 80 degrees for gene expression study. RNA extraction and the cDNA complementary DNA conversion. The liver was total RNA was extraction from the fish liver using TRI reagent, and actually we follow the manufacture protocols in this study. The purity and the quantity of total RNA were measured immediately by nanodrop, while the absorbance at a 2060 and 2080 ratio of above 1.9 was used to subsequent analysis. For this first standard DNA conversion, high capacity complementary reverse transcription kits were applied. We actually obtained the cDNA product was stored at minus 20 until analysis. The quantitative real-time polymerase chain reaction, qPCR, was carried out in a thermal cycler, and the primer sequence for the stress response genes, which is 4 genes, it's HSP70, NRF2, CYP1A, and AHR, and the 7 genes for the antioxidant genes, SOD1, SOD2, CAT, GSTP1, GBX1A, GSS, and GCR type. Statistical analysis. The data were submitted to two-way ANOVA to determine significant difference in MRA expression level between the control and treated group in the different exposure type. So the results actually we summarize in figure 1, as we can see now. In all elevation times, the 7 days, 14 days, and 21 days, the first 2 principal components cumulatively explained more than 80% of the total variation. Furthermore, the loading plot, which is showing the relationship between the PC, the principal component or the parent component, and the original variable, demonstrated that after 7, 14, and 21 days of exposure to pollutant, all hepatic genes evaluated were negatively correlated to the BC2. In this figure, actually, it's explain the relative RNA expression genes for A, the gene HSB70, B, CYB1A, C, AHR, D, NRH2 genes, all of them showing down-regulated and gene expression, especially after 14 days of exposure of all compounds treated in this experiment. In figure 3, it's shown actually and presented the relative RNA expression for 4 genes. A displayed down regulation for SOD1, C, CAT, D, GBX1A, and all the genes actually has been down regulated, especially after 14 days of exposure compared to the 7 days and 21 days. In figure 4, exhibited the relative RNA expression genes and demonstrated for A down-regulated for gene GSP1, B GSS, C GSR, and all of them too are down-regulated, especially after 14 days of exposure of the compounds that been tested. In this figure, actually, we show the cluster analysis with emphasis on the formation of subgroup that performance of all compounds, the 3 compounds we are using in this experiment. After A, which is 7 days of exposure, B, after 14 days of exposure, and C, which is 21 days of exposure, especially at concentration of 3 microgram per liter. In figure number 6, we did heat maps, which presented in this figure demonstrated that general gene expression in zebrafish exposed to the compounds EHMC and 4-MBA was more acculturated in the longer exposure, especially after 14 and 22 or 21 days of exposure, which is completely different from the 2EH exposure group. And we can see between the colorful figure from from C and B compared to the compound EH, which is D. However, the expression actually of the genes evaluated was more balanced in the different days of exposure, especially for figure B and C compared to figure D. The summary actually of this study, the impact of ethylhexyl methoxycinnamate and its photoproduct were investigated in zebrafish, especially in liver organ. Principal compounds analysis in 2 ways, repeated measured were performed. Stress response gene expression level were significantly altered in hepatic tissue. The 3 compounds, the EHMC and 2EH and 4-MMBA, exposure affects the antioxidant defense mechanism of zebrafish. And finally, the photoproducts showed similar effects than parent component. And the last point actually is really important. So the conclusion of this study explains that our findings confirm the hypothesis that an environmental relevant concentration of EHMC and its photoproducts alter the transcriptional level of stress response and antioxidant gene in zebrafish liver, and actually sheds the light on how this compound can affect freshwater and therefore reinforcement the danger associated with its dispersion in aquatic ecosystem, and the consideration that our study is not exhaustive, several other factors should be investigated. Finally, the effects of EHMC to AH and 4-MPA on gene expression in younger individuals or other fish species over long periods in association with other toxicity biomarkers will be useful to burden and knowledge the extent of ecological level associated with this pollution. And we can see actually in the abstract as a graphical here that our sunscreen compounds that we are using in summertime, the whole world using it, can has negative impact on the marine life ecosystem. This study actually we published in a highly prestigious journal, Science of the Total Environment. The impact factor is 9.8 and the site score is 16.8. Of course, it's a Q1 in Scopus and Web of Science. And we published this study in 2022 with a group of Brazilian scientists under a big project funded from the Brazilian government. And it's actually interesting study and one of the grateful investigation on how other chemical that we are using daily and occasionally can affect the ecosystem around us. Finally, actually, I would like to thank everyone, and I would like to thank Dr. Ahmed Atif Salam, the founder of the MGPS, and all the people in organization committee, Dr. Abdullah Talat, Dr. Abdullah, Zainab, and all our friends there, Dr. Amir, Dr. Syed Abdul Monsef, of course, the president of Assyut University, Ahmed al-Minshawi, Dr. Gamal Badr, of course, the dean of our faculty, Dr. Adel Muhammad Mahmoud, the vice dean, Dr. Muhammad Saghir, and thank you very much for having me. Thank you.
Thank you, Dr. Muhammad, for the interesting lecture. And now we will start the discussion for this seminar. So you can write your questions in the chat room, or you can raise your hand and I will allow your mic so you can tell your questions. So anyone has a question? Any questions? Okay, I may start. Dr. Muhammad, these components, do you think that has effect on the meat fish when people eat this fish?
In what? Sorry, Ahmed, in what?
In the meat of fish meat.
Okay. Look, Dr. Ahmed, in our study, actually, we focus on the specific genes in the liver, and we tested it like to see the elevation of the activity of and gene expression of these genes. And to be honest with you, we didn't do any experiments on the meat and the quality and the quantity and the contents of the muscles and even the whole blood test actually. But that's really good question, and we can take it this point in our next investigation, and we try to see if there is any impact of this kind of compounds, especially showing actually a big and negative impact on the liver. So next investigation, we will focus on the effects on meat of the fish to see if there is any negative impacts or it doesn't have any impact. our investigation will show us how it looks like. So that's a really
Okay.
good point.
Okay, thanks. I think David has a question, so I allowed your mic, so you can please unmute yourself.
Okay, good evening, everyone. Thank you, Dr. Ahmed, for
Yeah,
an enlightening lecture.
yeah,
But I would like
you're
to
welcome.
find out two things. One, to better understand this lecture, I want to know the three components you tested in the zebrafish. Are these components from the uh, sunscreen materials used by humans? That is one. Are they from the sunscreen materials used by humans? If that is right. Secondly, is it possible that these three agents or these three chemicals tested would have a similar impact in the human liver or human cell as it had in the zebrafish? I don't know whether this question is clear enough.
Okay, thank you, David, for your two questions. The first one, yes, we have three compounds. The first one is the parent compound, which is ethylhexyl methoxycinnamate, and it has two photo products, two ethylhexanol and 4-methyl benzaldehyde. So we test the parent compound and the two photo products of the parent compound. And as I can tell you, the three of the compound, like the parent compound and the photo products compounds, actually has negative impacts on certain genes, as I can mention in my study and my presentation. So this is the first question, the answer of my uh, justification to your first question. The second question, we didn't do actually any specific study on human directly, but we tried to give like a model like zebrafish, which is a standard model by WHO for investigation. So I can tell, but it's not 100% sure. It could be has like negative impact on human, especially on the specific genes close to what we are tested here. And we actually tested two groups of genes, the stress responsive genes and antioxidant genes in zebrafish. So to be sure and to be 100%, is there any bad or negative effect on human? I can't tell right now, but I can guess, yeah, it could be some negative impact on human based on our study.
Thank you. Thank you.
Thank you. Any other questions? Okay, so I think we come to the end of the lecture. Once again, I would like to thank you, Dr. Muhammad, for this interesting lectures. I'm quite sure that we get benefit from this lecture and information. And I would like to thank all attendees. And please, we will issue a certificate of attendance, but at the end of all seminars. So if you would like to get the certificate, please register here in the link in the chat. And
Thank you
thanks for
very
all.
much, Dr. Ahmed, and
Thank
thank
you, Dr. Muhammad.
you for all the organization committee, and thank you all. Thank you very much.
Okay. And just to follow up by knowing the upcoming lectures next week in the website, you will find a table of all lectures and a book of abstracts. Thank you.